Hello, my name is Rob Durham.
And I'm John Chappell.
And you're listening to another episode of the Gyrolab Spin.
This is a very special episode.
It is indeed, John, a very special episode.
We have some special guest stars, which we will introduce shortly.
Great.
First, I'd like to introduce Frida. Frida, would you quickly introduce yourself?
Yes, thank you, John. My name is Frida Löthberg, and I work as an application scientist here at Joris. Been here a bit over a year now.
Excellent, thank you.
So the next guest on our podcast today is also from the field application team, and his name is Joris. Joris.
I'm Joris Venet. Hello, everybody. And I'm working in Gyros since the beginning of the year as a field application specialist for the customer in the French-speaking region in Europe.
Excellent. I never would have guessed that you were from the French-speaking region, but
I am.
Welcome to the podcast.
Yes, so we're here today to tell you about some strategies for measuring oligonucleotides using Gyrolab and some different assay formats that we've been testing for this purpose.
So a good thing to mention is that the 95% of the data that have been generated were generated by Frida for her master's thesis. So few key points about oligonucleotides are, so they are usually like 15 to 25 nucleotides length, complementary to the target mRNA, and unmodified oligonucleotides will be quite rapidly degraded in the body because there are many nucleases that are going to attack those single-stranded DNA.
Yes, and to combat this, there are several chemical modifications that can be done, both to protect from nucleases and also improve target affinity and deliveries to target organs. And also covalent attachments such as GalNAc is a common example, which would help with biodistribution and cellular uptake. So when you talk about oligonucleotides in a medical sense, there are very many different variants, and a few of the common ones are mRNA vaccines, antisense oligonucleotides, aptamers, and small interfering RNAs. And as of the end of 2021, there were 15 FDA-approved oligonucleotide drugs on the market.
So yeah, and there is now many more clinical trial. So that's why we have like the needs to be able to quantify those oligonucleotides, and they are mainly used for treating generic and infectious disease. Depending
on the different structures and sequences of these oligonucleotides, we have different modes of actions and delivery systems. Some examples are steric blocking. You can also do splice modulation, where you hide splice sites, or you can attract RNases to cleave certain mRNAs, for example.
Yeah, so there are different structural variants of those RNA and DNA. So the first at the left is the nucleotide without any modification. And then the first generation of oligonucleotide of azo is the phosphonate. Then the second generation is the 2-O-methyl and the 2-methoxyethyl version, and the third generation, which is the PMO, and we used this chemical structure to generate the data we are going to present you. So there are some important bioanalytical parameters that need to be considered. First, the high sensitivity of the assay is really important, depending on the matrix you are using. So you can develop an assay in plasma, in serum, or even tissue. You need as well to check really the specificity of your assay to the parent oligo, and you can, in that case, test some metabolite of the parent oligo to test if you are detecting them or not, and this in order to not have improper PK interpretation. So Frida, let's have a look to the different assays we developed.
Yes, absolutely. So comparing this to a classical sandwich assay, which most of you may be familiar with, we have the capture antibody, which is bioattenuated, and then a fluorescent detection antibody. The same principle is true for the hybridization assay. You can have two probes, one detection oligo and one capture oligo. And they're both, in this case, complementary to half of the oligoanalyte that you want.
So for this method, what we want is really to fully automate the process and don't use the bioaffy CD, but the mixing CD that will allow us to do the hybridization in the mixing chamber of the CD, and then the mix will go through the strepta column.
So we would first test that this assay works offline as intended, but then we would like to move on to the CD to have it fully automated to exclude all of the incubation steps that you would have to do online. We showed this format just a couple of slides ago, which is called the dual hybridization assay. So it utilizes two probes, just as a sandwich assay, capture and detect. Without the binding of the analyte to both capture and detect, the detection probe would be washed away. But this has the potential to detect 3' and 5' metabolites, as the hybridization could be strong enough to retain both.
So the first step of this dual hybridization will be the addition of the sample in the CD. So the sample is going through the mixing chamber, and then the capture probe is going to be added to the CD as well.
So these two are then mixed in the mixing chamber with the 1 minute mixing sequence, and then incubated for between 5 to 30 minutes, depending on how long you want to have it there. So they would then hybridize in the mixing chamber.
So in that case, here the capture reagent is going to hybridize for the first half of the analyte, and then the second reagent is going to be added in the same inlet, and then going through the mixing chamber to be mixed as well with the capture and the analyte. And here the detection reagent is going to bind the other half of the analyte.
Yes, so we then have a fully formed complex that would run through the streptavidin column and hopefully be caught by the streptavidin beads. And this can then be detected.
So another method that we developed is the nuclease-dependent cutting assay. So in that case, we are using a single dual-purpose probe. This probe is going to be biotinylated at the 3' end and is having a fluorescent dye at the 5' end, and will bind the analyte on the full length. And then a nuclease can be added, and this nuclease will be selective for single-stranded DNA and will cut any single-stranded DNA to be able to detect only the analyte that is bound to the dual-purpose probes. So this method is supposed to be selected for the full-length analytes, so it's supposed to be really specific for the parent oligo.
In this case, you would add the full-length capture probe to the mixing CD, which would then go through the volume-defining chamber and down to the mixing chamber. And then you would add the analyte, which would hybridize with the full-length probe in the mixing chamber. You would incubate this for 60 minutes.
So and then you are here then producing the double-stranded DNA with the sample that is hybridizing with the dual probe, and this mix is going to go through the streptavidin column. Here the streptavidin column will bind the double-stranded DNA, but the single-stranded DNA as well, and if we are not adding any nuclease, then the signal will be like really high everywhere, because the single-stranded DNA will give some signal.
Therefore, we add, in this case, micrococcal nuclease, which we will run through the column, wouldn't incubate it with the samples. And this will cleave any single-stranded probes and remove the fluorescent dye, so we will only detect the hybridized complexes. Working with this assay, we had two workflows. Firstly, the manual assay, where we use Gyrolab for detection, but all of the reagents and mixing and incubation is done outside of Gyrolab manually. And then we're running a one-step assay on a bioassay 1000 CD just for detection of the complex.
And then the automated assays, which is using the mixing CD we just talked about. So you have the 20 minutes reagent preparation, and then you load everything in the gyros. And depending on the incubation time you are choosing for the reagent with the analyte, your run can be like 1.5 hours to 3 hours, but it's really fully automated 3 hours, so you don't need to do anything during this.
And here we wanted to compare the manual assay to the automated assay and see if we could get comparable results.
Perfect.
So let's talk about the result we generated on the dual hybridization assay.
For the manual assay, where we did all the reagent mixing and the incubation outside of the CD and the Gyrolab, we had 2 times 30-minute incubations. So we incubated the capture reagent and the analyte for 30 minutes, and then added the detection reagent to that mixture and had another 30-minute incubation. And then we ran this mixture through a bioassay 1000 CD with a one-step assay just to detect the hybridized complex. And we saw no difference between incubating the hybridization reaction in room temperature or in 37 degrees Celsius.
So yeah, this kind of results are pretty good for us, because if we want to move to the mixing CD, we have no choice to work at room temperature.
When automating this assay, we kept the same format as we did offline. So adding capture probe and analyte to the mixing chamber with a 30-minute incubation, and then the detection probe for another 30 minutes, and then running it through the column.
So here you can see in the graph that we tested as well different incubation time, coming from 30-minute to 5-minute incubation. What we can see is that we are losing a bit of signal when doing like smaller incubation time, but this is something that can be kept in mind, because then depending on the oligo you are using, the hybridization will be different.
Yeah, and we also saw a bit lower signal to background ratio with the lower time incubations, but nothing significant.
We had the possibility as well to test different chemistry of our oligonucleotides. So first, all the data were generated with the PMO chemistry, and we tested as well the same protocol using the 2-methoxyethyl chemistry. You can see the results are pretty much the same. The sensitivity is really good, and the dynamic range are quite equivalent for both. But we could consider that if we want to use the 2-methoxyethyl, we could optimize a bit the assay for having better precision, for example, at the low part of the curve.
Continuing then with the PMO chemistry, we started working with serum. And so the standard curves you can see on the right are 50% human serum. We also had QC samples that were spiked in neat and then diluted 1:2. And from 6 runs with this configuration, we calculated an LLOQ of around 2 picomolar in neat serum. As we talked about earlier, we wanted to test the metabolite cross-reactivity with the parent oligo. So we used 6 different metabolites. We had up to 3 nucleotides removed from each end, the 3' and the 5' end.
So each of those 6 metabolites were tested at different level, and for a perfect specificity, you will recover 0% of signal. But here, in that case, as you can see, one of the metabolites is showing 40% of cross-reactivity for the N-1 at the 5' prime.
And this is still quite surprising, since with this assay format we had expected a lot more cross-reactivity across all of the metabolites. Now we will be moving on to the results of the nuclease-dependent cutting assay.
So as for the hybridization assay, we first assessed the manual assay, doing all reagent mixing and incubation outside the CD. So here the dual purpose probe was incubated with the analyte, and then the micrococcal nuclease has been added to cut every probe that is not bound to the analyte, and the micrococcal nuclease has been added at 10,000 unity per milliliter, and we again assessed the incubation at room temperature and at 37 degrees, and quite same results were obtained again here.
Yeah, and it would be even more surprising here, since in the literature it states that micrococcal nuclease works the best at 37 degrees, but this is fortunate for our automation. During our automation attempts, we ran into some problems here with the replicate variability, and it seems that the buffer that's compatible with the nuclease isn't necessarily compatible with the microfluidic system. So in a buffer with low or no salt, we have very high replicate variability, but when increasing the high salt concentration to remedy this, we lose sensitivity instead, since the nuclease doesn't tolerate high salt concentrations.
So yeah, and then using an optimized salt concentration buffer, we can reach a really good dynamic range and a good sensitivity, which is mimicking the data we were generating in the manual assay. So then, in order to still improve the assay, so the precision of the assay, we screen different Rexy buffer at different dilution, and here the two that are presented here, so the H6 and the CCS, were giving the best precision data.
So we chose to continue using Rexip CCS at 100%, also mixed with nuclease reaction buffer and some BSA. We also tested adding 10% serum to the analyte just as the stabilizing agent, but this didn't have any effect on the assay.
So as for the dual hybridization assay, here we tested the specificity using the 6 different metabolites, so the same one, the 3 on the tri-prime end and the 3 on the 5' end. And here we are obtaining a bit more cross-reactivity with the metabolites on the 3' end. We have like 25% recovery against the 10% on the 5' end.
And this could be explained with that the micrococcal nuclease is supposed to generally be more active on the 3' end, so we would have an easier time cutting there. But we would also have expected lower recovery in general with this assay format, since it's supposed to be more selective.
Perfect.
And now to conclude. This slide is an overview of the methods that we've used, the micrococcal nuclease assay. We're also showing the S1 nuclease, which was used in the same format, but we haven't presented the data here.
And we have as well, so in green, the dual hybridization assay, which is giving the best sensitivity and the best dynamic range. Here the specificity for this assay was surprisingly quite good, which is maybe due to the Gyros automation.
Now our conclusions here for the dual hybridization assay, it works really well on the microfluidic system. We of course want to test more sequences, since we only work with one, and possibly more chemistries as well. And the metabolites have considerably lower cross-reactivity than has been reported and what we would have expected, so this is really nice.
Yeah, and about the nuclease-based assay, we had some trouble at the beginning to deal with the precision of the assay due to the salt intolerance of the micrococcal nuclease. But in the end, with the CCS Rexy buffer condition, we improved a lot the assay, and we tried as well other nuclease that could make the assay a bit more sensitive than what we have. For the specificity, the metabolites show a low cross-reactivity, but still a bit of cross-reactivity.
Yeah, so in this case, we hope that the DHA assay could be acceptable on the Gyros format, and we might not need to work with the nuclease, since the cross-reactivity is so low, and that's was supposed to be the main advantage of the micrococcal nuclease assay.
So we are done with our presentation. So we were glad to present you the data we generated, and we thanks John and Rob for giving us the possibility of presenting this here.
Yes, it's been a great time being here, and thank you everyone for listening. And John and Rob, do you have any questions for us?
Yeah, great job, guys. I've got one question for you. Many people that are doing oligonucleotide assays are using digoxygenin as a labeled detection probe. Did you have any experience using that, and how did it compare?
Yeah, we tested using digoxygenin labeled probes and then detection antibody against that. But we got much lower sensitivity than we would the directly Alexa labeled probe, so we continued with those.
Great, and the other thing about your work that I really like was you had relatively low cross-reactivity with the metabolites, but that may be sequence specific. So I was wondering if you encountered some more metabolite interference, what would be your alternative? Any thoughts on what you would do to help combat that?
Yeah, this is an excellent question, and we were thinking about it. And as the Gyros is a really open platform, we have the possibility to add some steps after the dual hybridization method, and we were thinking to add a nuclease wash at the end after that the mix is going through the column in order to decrease the signal from the metabolites. We could as well try to reduce the incubation time in the mixing chamber to reduce the specific binding from the metabolites.
So that brings us to the end of another podcast, John.
Yeah, no, it's an excellent job by these guys. It was good to hear this scientific area. It's actually an area which we've seen a lot of interest with, and I think a lot of people are interested in oligonucleotide assays on our platform. But just as a general point, if anybody's got any potential areas for us to cover in these podcast series, please contact us. The email addresses are shown. Also, we are likely to include further guest stars going forward, potentially customers and also other scientific experts to give an introduction to a scientific area on the platform.
Yeah, I mean, I get tired of just looking at John's face all the time.
Yeah,
I'm sure he feels the same about me. So it's fun to have some new faces on the podcast.
Yeah, especially younger faces.
Yeah, younger faces, correct. Well, that wraps up the podcast for today. So
It's a goodbye from me.
It's a goodbye from her.
and it's a goodbye from me.
And it's a goodbye from him.
And until next time.